Hello and welcome to my video about mitosis. In this video, not only will I be teaching you mitosis, but I will be accompanied by little pictures which at the end of the explanation will be combined into a stop motion film. All the color coding is in the description down below, and I really hope you enjoy this video. The first phase we'll be discussing in this exploration of mitosis is interphase. Now, interphase is not exactly a part of mitosis, but it's still a really important process to the cell cycle. Interphase can be summed up by just growth in a cell. As you'll see here, after a whole cell is completely drawn, that the centrioles will replicate and the DNA also will replicate, and that's what determines interphase. Next phase we'll be discussing is prophase, and it marks the official intro into the actual mitosis process. Two important events happen in prophase, the first being that the chromosomes begin to condense and they start forming chromatids, which is pretty much just one copy of a duplicated chromosome. The other thing that happens is that the centrioles start coming closer together and forming an early mitotic spindle. Now that we've discussed the things that make prophase, we can move on to the next phase, which is pro-metaphase. Two major events define prophase, the first being that the nuclear membrane begins to break apart. Eventually it completely breaks apart. The second being that the microtubules, which are represented by the little lines, they start attaching to the kinetochores, which are just proteins that are on the chromatids that allow the spindle fibers to attach to them. Pro-metaphase is complete, which I would like to emphasize is mainly the nuclear membrane breaking apart. We go into, you guessed it, metaphase. Metaphase is mainly defined by only one thing. Pretty much in metaphase, all the chromatids line up in the middle at something called the metaphase plate, which is surprisingly not greatly represented in these pictures, the straightest line in nature, or one of the straightest lines in nature. After everybody is lined up in the middle and we're done with metaphase, we go into anaphase, which is also my favorite phase. The reason why anaphase is one of my favorite phases is because it's relatively short. The chromatids separate and that's it. After we leave anaphase, we go into telophase, which is defined by a lot of things. One of the first things that can define telophase is that the cells begin to elongate. This is because they're ready to separate, so they actually are being pinched into the middle, which creates something called a cleavage furrow, which is represented in the drawing by the black line in between them. They, as you can see also, the daughter nucleuses, which is just pretty much the new chromosomes, have a nuclear envelope which is created by the old nuclear envelope. After telophase is over, we finally reach the last phase, which is called cytokinesis. Cytokinesis, what we'll be doing this whole time, finally pays off and the cell breaks off into two identical daughter cells. Thank you for watching my video. I hope you enjoyed it, you found it informative, and that it somehow helped you. If you have any questions, you can leave them in the comment down below. You can check the description box for more information, and I just hope you like the video and enjoy the film that will be played after this, which is just the images all together.